All right, what's up, Honors Bio? This is Mrs. Kozian again. This is going to be Unit 2, Biochemistry, and this is Part 4, Enzymes. You guys, there's all kinds of really cool research out there about enzymes right now. You might have heard some ideas about, enzymes being a part of research into celiac disease. If you don't know what celiac disease, we can talk about it in class. Crohn's disease. And there's even some research out there about digestion and enzymes in autism. So there's a lot of interesting questions that you might have about this, and make sure you write them down as you write down your notes. All right, things you're going to see here, you're going to see or things you might see in this unit, the definition of the word enzyme, what a catalyst is, activation energy, an enzyme called lactase, and then a Benedict's test. Those are both things that we are going to do in lab, lactase and the Benedict's test. You guys also might see the word substrate and the word active site. Those are words that you're going to see in this unit, these notes. All right, so start with the word catalyst. It's a substance that can speed up a chemical reaction. I always think of the word catalyst. I remember hearing it in like literature class when I was in high school when we were reading Romeo and Juliet, if you guys have had to read that yet. And I remember different things in the plot, my teacher calling them a catalyst, like they were a catalyst for the plot or the story, and they were things that speeded up or changed the story. So catalyst is anything that's going to speed up a chemical reaction. That can be heat, that could be a specific pH, that could be stirring or shaking. It could be, sort of mechanical mixing. Another thing that is a biological catalyst are enzymes. Enzymes help to speed up chemical reactions for us. All right, and here's an enzyme right here. It's a protein that's going to speed up a chemical reaction or act as a catalyst. Remember, catalyst speeds something up. Enzymes have an ACE ending, ASE, and the name of your enzyme will come from the reaction that is catalyzed or the reaction that's sped up. So for example, lactose, we're going to talk about lactose in the milk lab. The breaking down of lactose is helped by the enzyme lactase. So you can see breaking down of lactose is sped up by lactase. So that's why it ends in ase. Okay, so we take the reaction or the protein or carb or whatever's being broken down and add an ase to the end of it, and that's usually the name of the enzyme. Example here. All right, this graph, the brown line is representing the energy that's required for the chemical reaction when there is no enzyme present. If you guys can see, this is activation energy. This is the amount of energy it takes for the chemical reaction to happen. And when we don't have an enzyme present, there's a lot of energy. It's kind of like a big hill that we have to get up. But when we add an enzyme, it helps lower that activation energy, makes it easier and faster for us to finish this reaction. Okay, so here's what the reactants we start with, carbon dioxide and water, and then we end up with a product. Okay, here's just a chemical reaction example. So when we add an enzyme, it's a lot easier for our body to do that. And the definition of activation energy, it's the energy that is needed to start a reaction. So the energy to get up that hill that you saw in that graph. Enzymes make that hill smaller or make the activation energy that we need lower. Chemical reactions require breaking bonds and reactants and forming bonds and products. Remember, we're going to change our sort of our definition of where energy comes from. We know that energy is in bonds, and anytime that we have to process anything in our body, we end up breaking and making bonds throughout that process. So what an enzyme does is just makes that process quicker for us, makes that process much quicker for us. We'll have to break some of the reactants and then make new ones in the products. Some reactions are way too slow. We need enzymes to lower the activation energy. Think about a lot of the reactions that happen in digestion. If we had to just wait for food to digest on its own, think about, it could take days, weeks, months for food to break down if we didn't have enzymes to help us to do some of those things. And we also have bacteria in our digestive system, but the bacteria very often use enzymes to help us to make those macromolecules, to break them and make new ones. So activation energy, energy we need, and if we have enzymes, we need less. All right, here's just a couple graphs to show you what happens when an enzyme is present. So it shows here energy yield. This is the energy we need from beginning to end. Okay, so you can see here's the energy we start with, here's the energy that's at the end. And the energy yield is the total energy that you're going to need to finish the reaction. Now what a enzyme is going to do, it's going to help us to get to that quicker. Sorry guys, again, I hit the back button a few too many times. All right, I've got activation energy. We went over this graph already. Just another little representation. You can see the first picture is no enzyme, and there's a lot more energy required in order to make the reaction happen. Here's the products. You can see the frogs turning into princes. And when we have an enzyme, it lowers the activation energy so we can get more product with less energy required to get it started. All right, now enzyme substrate complex. You guys will need to know this information for your test. A substrate are the reactants of the reaction, so they're the thing that we need to process. And what happens is the enzyme comes in and binds to that substrate in what we call an active site. It acts like a lock and key. Certain enzymes will only process certain substrates. They fit together very specifically. So you can see here, this is the substrate, this is the stuff that I want to process, and this is the enzyme that's going to help me make that happen. This enzyme, obviously you can see the active site doesn't fit, they're not like a lock and key, so this substrate can't be processed. Okay. But when we bring in this is an allosteric activator, it's going to help us to make the substrate fit into our enzyme. When our enzyme and substrate fit together within that active site, then the substrate can be processed. Okay, so we call that the enzyme substrate complex. When the substrate correctly binds to the active site in an enzyme, we can then go through the reaction. We can come out with the product that we want. Okay, reaction happens, they stay together until the reaction is complete, we get products, and then the enzyme releases the products, and then it's open to react again. You guys need to know the steps of the enzyme substrate complex for your test. You guys don't need to know allosteric activator. You just need to know that the enzyme and the substrate fit together, they go through a chemical reaction, and then the substrate's released and the enzyme can react again. Enzyme regulation. So enzymes, because they have specific jobs, can only work in really specific conditions. Okay, so they won't do their job unless they're in the right place. For example, enzymes in your saliva work under specific pH levels and temperature. They won't work in your stomach. There's different enzymes that work in your stomach, and then there's completely different enzymes that work in your intestines. So they all are very, very, very specific in their jobs. Just like, say, you know, every teacher is very specific in their job. I don't teach English, you know, I don't have any English teachers teaching science right now. So anything that could change a chemical reaction can also change enzyme activity. pH values. You can have certain enzymes that work in really acidic areas like your stomach, and then other ones that work in more neutral places like your saliva. Also temperature. We have different enzymes that work at different temperatures. For humans, our normal body temperature in Fahrenheit 98.6, you may have heard, is equivalent to about 37 degrees Celsius. So we have several enzymes that really like to work at that temperature, and then others that work at, you know, lower or higher temperatures within our body. All right, your cells can also turn your enzymes on and off with the production of protein at critical stages. Your DNA is the one that tells us how to do that. Now, sometimes if we have a DNA mutation or an altering of an enzyme, it could be because we don't have maybe the correct directions in our DNA. And you guys can research some of those for your current event if you want. Example of cells turning enzymes on and off would be enzymes in your Siamese cats. Yes, you might see, if any of you have a Siamese cat or have ever seen one, they have a darker face, darker ears, darker paws. And it creates more melanin in colder parts of the body. So when we create more melanin here in the face and the ears, sorry guys, I hit the mouse too many times. Okay. And then it's less active in warmer parts of the body, so that's why the rest of the cat actually is a lighter color. So it's an enzyme that's gonna work at a specific temperature. So it's just one example. Cells can turn it on and off at specific in specific environments. All right, another enzyme graph example. You guys can see the red line is the energy required without the enzyme, and then the blue line is with the enzyme. Again, lowering that activation energy. Make sure that at the end of these notes you understand what type of macromolecule is an enzyme. It's a protein. and enzymes can impact a reaction by helping to speed them up. Make sure you can explain why the enzyme substrate complex is compared to a lock and key, because specific enzymes only work with specific substrates, and make sure you can explain what happens in that complex. And then some factors that can impact reaction rates, like pH, temperature, anything that's going to affect a chemical reaction is going to affect the reaction rates with an enzyme. All right, and that is the last of your notes for this unit. Woo! Good job, honors.